So the last time we left off talking about Chapter 13, bioenergetics and reactions, and we're going to follow up talking about some specific types of transfer reactions and reactions that we see with highly favorable delta G values. So one of the most common transfer reactions that we see is the phosphoryl transfer from ATP. So you'll notice that if I transfer one phosphate group from ATP, I'm going to form ADP, as well as some type of a phosphate ester to whatever molecule I've transferred that phosphate group to. And if I'm thinking about the reason why I use ATP, it's highly favorable under standard conditions. So there's better separation in the products, I have better salvation of my products, and I actually have favorable resonance stabilization of the products. So these are the three primary reasons why ATP is so prevalent as a molecule for transfer. So one of the things that's interesting here is that the charge separation that results from hydrolysis is really what relieves the electrostatic repulsion. And if I think about inorganic phosphate, that's going to be stabilized by resonance, where essentially I can move that double bond to each of the oxygens with the same degree of double bond character, right? So that charge is distributed. And we also recognize that ATP hydrolysis has a greater degree of salvation or hydration relative to ATP, which also stabilizes the products relative to the reactants. When I'm thinking about the actual free energy change in a process, I know that that's going to depend on energy. It's also going to depend on the actual concentrations of the reactants and products. So free energy change is going to be more favorable if the reactant's concentration exceeds its equilibrium concentration. So again, think about that K equilibrium value and the fact that you want your products to be larger than your reactants in terms of concentration. So true reactant and the product, right, are ATP and ADP respectively. So if I look at delta G, I know that that's going to be dependent upon magnesium. So I could write the following expression for the overall Gibbs free energy. The reason why ATP hydrolysis is dependent upon magnesium 2+ ions is because of the coordination. So you can actually see that here for both ATP and ADP. If you look at table 13-5, this shows you hepatocytes, myocytes, neurons, erythrocytes, and E. coli cells and the relative concentrations of ATP, ADP, and AMP. So if you think about this, cellular ATP concentration is usually going to be very far above the equilibrium concentration. So ATP is going to be a very potent source of chemical energy in virtually all cells. If you look at phosphorylated compounds in general, they're going to have a large delta G for hydrolysis. So if you think about electrostatic repulsion, that makes sense. So by doing hydrolysis, you're relieving electrostatic repulsion. And because the products are stabilized via resonance or by more favorable salvation, you can actually also potentially see further tautomerization, right, to form the keto versus enol form. And that too can contribute to a larger delta G that's negative. If we look at phosphates and we rank them by the standard free energy of hydrolysis, you can transfer phosphates from compounds with higher delta G to those with lower delta G. So for example, reactions such as the phosphoenol pyruvate plus ADP can form pyruvate plus ATP. Those are going to be favorable and can be used to synthesize ATP. And again, right, if you think about your phosphoryl groups, it's really just the ATP to the acceptor molecule, such as glucose or glycerol, that forms these low energy phosphate derivatives, right? And again, we can also talk about that in terms of the hydrolysis of thioesters, which is strongly favorable. For example, something like acetyl-CoA. So acetyl-CoA is an important donor of acyl groups. It is feeding 2 carbon units into metabolic pathways, and it's the synthesis of fatty acids. So in acyl transfers, molecules other than water are typically what accept the acyl group. Here's an example of what that transformation looks like. So acetyl-CoA is a thioester that has a large negative standard free energy of hydrolysis. Thioesters contain a sulfur atom in the position occupied typically by an oxygen atom in oxygen esters. And if you look at the complete structure of coenzyme A, you can find that in figure 8-38. You're going to notice that the business end really is this acetate. So if we think about why these thioesters might be reactive, the reason for that is the orbital overlap between the carbonyl group and the sulfur. That's not going to be as good as the resonance overlap between oxygen and the carbonyl group in esters. So while the contribution of ATP to a reaction is often shown as a single step, it's almost always actually a two-step process. So here, if we're taking a look, this is a reaction that's catalyzed by ATP-dependent glutamine synthetase, and the phosphoryl group is transferred from ATP to glutamate. Then the phosphoryl group is displaced by the NH3 and released as a free phosphate. The last thing we're going to talk about is oxidation reduction reactions. So these are reduced organic compounds that serve as fuels. So you can strip electrons during oxidation and shuttle them elsewhere. Many biochemical oxidation reduction reactions involve the transfer of 2 electrons. So in order to keep the charges in balance, proton transfer often accompanies electron transfer. So in many dehydrogenases, it's the reaction that proceeds by a stepwise transfer of protons and hydrides. So for example, in the conversion from lactate to pyruvate, and using the enzyme lactate dehydrogenase, it's the transfer of 2 electrons accompanied by the transfer of 2 protons. So if you're thinking about reduction potential, then you should be able to determine the affinity for electrons. So the higher the E, or the reduction potential, the higher the affinity. And these electrons are transferred from lower to higher E. So in the expressions for E shown here, you can actually calculate what the overall delta E would be. And again, in order for a negative delta G, you need to have a positive delta E. This is a list of standard reduction potentials of some of the biologically important half reactions. These are reduction potentials that you're going to be using in the problems that we're solving in class. When we think about NAD and NADP, these are common redox cofactors. They're called pyridine nucleotides because they can dissociate from the enzyme after the reaction. They're incredibly powerful. In a typical biological oxidation reaction, the hydride from an alcohol is transferred to NAD+, resulting in NADH. If you take a look at the reduced NADH, you can actually see here the transfer of those 2 hydrides. The formation of NADH can actually be monitored by UV spectrophotometry. So you can measure the change of absorbance at 340 nanometers. This is a useful signal when you're studying the kinetics of NAD-dependent dehydrogenases. It's actually one of the ways to be able to measure the activity of an enzyme. Flavin cofactors also allow single electron transfers. So in this case, that's the use of molecular oxygen as the ultimate electron acceptor. So these are flavin-dependent oxidases. The cofactors are tightly bound, however, to proteins, which makes them distinct and different from NADH. And so when we're thinking about the structure of FAD in its oxidized form, the flavin nucleotide accepts 2 hydrogen atoms, which is why you form FADH2. Right? So 2 electrons and 2 protons, both of which appear in the flavin ring system. So when FAD or FMN accept only 1 hydrogen atom, the semiquinone is a stable free radical that also can form. We're going to leave it there. Thanks so much. Have a great day.